I am a postdoc in the lab of Dr. Catherine Haig at Rocky Mountain Laboratories, which is a branch of the National Institute of Health. So today I'll be talking to you about a project that I've been working on. I will say a lot of this data is very preliminary, but it's modeling a new model for studying sporadic CJD. So there are quite a few difficulties in studying sporadic CJD. First of all, the cause of sporadic CJD remains unknown. And also, people don't know that they have sporadic CJD until after their symptoms have appeared, which is unfortunately after irreversible neuronal loss has occurred. Most mouse models and cell culture models don't actually develop CJD spontaneously. Instead, they need to be infected, which makes it not exactly the most accurate representation of the disease. And obviously we've seen a million amazing mouse models today, so I'm not trying to say that those are bad. I think that we should definitely take all of the approaches that we possibly can to targeting these diseases, but we do know that mice don't always recapitulate what is seen in humans, both in their clinical manifestations as well as their response to treatments and such. So this really leads to our lab's overarching question: can we develop a new research model for studying sporadic CJD? So my lab focuses on the organoid model, which many of you may have gotten a little bit of background information on yesterday, but I'll just kind of walk you through how this looks in the laboratory. So we got a generous donation from Dr. Wenquanju of a bunch of fibroblasts from different patients, both people who are healthy, people with genetic disease, and people with sporadic CJD. So these are skin biopsies that were then grown in culture. So these fibroblast skin cells were sent to us, and we have a small library of them. And from those, I've been able to take some of these fibroblasts, specifically ones from a patient with MM1 and ones from one with MM2 sporadic CJD, and reprogram them. So basically treat them with these different genes that cause the cells to revert back into what they were when they were stem cells. So these are called induced pluripotent stem cells, or iPSCs. And they have a lot of the characteristics of stem cells, where they can differentiate into a huge variety of different cell types in the body. So of course, we're very interested in neurons because those are what are actually killed by prion disease. And then we also wanted to develop these organoids, which are kind of like mini brains. So organoids we can grow in culture. They're about the size of like a very small pea. And they start out as being neural stem cells, and then they start to develop more mature cells. So by the age of 4 or 5 months in culture, they have a huge population of all sorts of different types of mature neurons that actually can function like normal neurons in the brain, interact with one another, and then they also have support cells such as oligodendrocytes and astrocytes. So I'm just going to show you some of my preliminary data. These are very long experiments because we start out with the organoids being first looked at about 4 months old. So the reason we choose this time point is because that's when they actually show significant population of mature neurons, as well as those helper cells, the astrocytes. And it's important to note also, I forgot to mention this, that my lab has actually taken these organoid models and just ones from healthy patients and infected them, treated them with brain homogenates from sporadic CJD patients. And we do actually see the development of prions in these organoids, and we see different signs that might be similar to what you see in an actual human brain. So that's pretty cool. But I've taken these organoids out to about 8 months, so this is kind of a longer project. But at 8 months, in a healthy organoid, so one that was from a healthy donor, we see this is brown staining here is MAP2, which is a marker of mature neurons. And here, this is kind of a visual representation of what neurons look like when they're breaking down. So in a zoomed in version, we can actually start to see little like cell bodies of these neurons here. And I'm not a clinician, I don't look at this stuff, but to me, this looks like a very healthy brain, like what you would see in a healthy brain. However, if we look at this 8-month-old sporadic CJD organoid, we see something a little bit different, which is kind of these like little patchy blotches, which to me looks kind of similar to this visual cartoon representation of what you'd see in neurodegeneration. So these are like really preliminary findings from like a couple weeks ago, so we still have a lot of work to do. But we also have seen this kind of thing in some of our organoids that we've infected with sporadic CJD, so we hope that it could actually be a real result. So this is every researcher's favorite assay, which is the Western blot. I think you've already seen a few of them today. But basically what this is showing is each of these bands is a different organoid. So these ones are from a healthy donor, these ones are from a sporadic CJD donor, and these are 6-month-old organoids. And what we can really do is look at the amount of protein expression, relative protein expression, in each of these different blots. So these top 3 here are looking at different markers of neurons. It's not super important to know what these are, but these should be expressed in a healthy neuron. And these bottom 2 are different markers of astrocytes, which are those like helper cells in the brain. So when we look at healthy and sporadic CJD organoids at 6 months, we don't really see a whole lot of differences in the relative amounts of protein here. However, when we look at the 8-month organoids, it shows a different story. So the healthy ones remain pretty similar in their amount of expression to what we see at the 6-month-old, but the ones from the sporadic CJD donor start to show a huge decrease in some of these neuronal markers, PSD95, NFL, and NUN, when we compare to the healthy 8-month-old organoids. So you might see this and think, okay, maybe these organoids are just dying off, but we also still see significant amounts of astrocytes at these time points. So if they are dying off, it might be specifically the neuronal population that is dying off. So ultimately, we see decreased protein expression from our neurons, but no real changes in the astrocyte markers. So hopefully I've convinced you that sporadic CJD organoids, as well as neurons, which I didn't show the data for today, can be used to study neurodegeneration. We successfully reprogrammed cells from a donor with MM1 and MM2 sporadic CJD. The data I showed you today was from the MM1 donor. And so far, none of these organoids or neurons have shown any sort of prion seeding activity when we've looked at them with RT-Quick. So we don't actually know if there's any infectious prions in there. At this point, we haven't seen that yet. And so we do know that the organoids do seem to show some neuronal loss as they age. And then I've also seen functional abnormalities in the neurons from these sporadic CJD donors compared to healthy neurons. So there's a lot of future directions here, like I said, very preliminary. I'd like to continue reprogramming cells from other donors and be able to study different types of sporadic CJD. I'd also like to continue monitoring neurons and organoids for prion seeding activity, and then, of course, test therapeutics to see if we can actually reverse this neuronal dysfunction and neuronal loss. So it's really been an honor to be here today. I'd like to say a huge thank you to the donors for supporting this work, specifically the Carol Meltzner Memorial Grant contributed by the CAS Foundation. And I would love to talk to any of you guys individually later and answer any questions. So thanks a lot.